speaker, who is Gary Wong. Gary is the head of the virology department at the Institut Pasteur de Laos, and he will be speaking on the environmental genomic surveillance of avian influenza A viruses in high-risk live bird markets in Laos. Gary, welcome to the stage.
Good afternoon, everyone. It's a big honor for me to be here to be able to present some of our work done in the last year on avian flu. So we'll get right into it. Avian flu, just for those of you who don't work with viruses, they can cause severe disease in birds and humans, respiratory disease. The case fatality rate for some infections with some subtypes of avian flu can be up to 70%. So bird flu can be carried by wild birds or domestic poultry, and typically we can classify them as HX and Y. So you may have heard about H5N1, H7N9, H9N2, and so on and so forth. And most countries in the world, so if you look at continents, you look at North America, Africa, Europe, Asia, and South Australia have reported an outbreak, at least one outbreak of bird flu in their bird populations. So in Southeast Asia, if you're local in Southeast Asia, typically if you want to source chicken to eat, you will go to what we call live bird markets, so LBMs. And this is a very common way to buy and consume live fresh poultry. Many of the species of these poultry, geese, ducks, and chickens, are sold in a confined area. And these birds may carry avian flu, such as H5N1, H9N2. And so because when we go to these markets, so whether you're a vendor or a customer, you may have close contact with these infected poultry, and you may, and the virus may spread to you via this mechanism. And actually, in Southeast Asia, so if we look at Cambodia and Vietnam, which are two neighboring countries, many cases of these influenza virus infections have come through direct contact with poultry. So this is why it's important to monitor what bird flu is present and circulating in these private markets so that we can detect them before they spread to humans. And so with the support of IPSN, since November of last year, we implemented a pilot genomic surveillance program in these live bird markets. So we chose two locations. So one is in, if you look at the two red dots on the right-hand side, in northern Laos, we have Luang Prabang, and in central Laos, so the bottom dot, we have Vientiane. So we chose live bird market in these two cities, and what we do is we go every month and we collect 60 samples, so 20 animal samples and 32 environmental samples at each site. And the environmental sampling can include air, wastewater, slaughtering equipment, so on and so forth, feces, and animals are just direct swabs, oral and chlorical. And for samples that are AIV positive, we try to generate whole genome sequences using nanopore technology. And then once we have the full-length genome sequences, we wanted to see the prevalence, like where the hotspots for AIV are in these markets, and what kind of mutations these sequences have that may make it more dangerous or transmissible or et cetera to humans. So why do we collect different samples? So as you can see, we do direct swabs, we swab the environment, and we also more recently started doing air sampling. And the reason is because we want to minimize the chance of not detecting AIVs in these live bird markets. Because we swabbed animals directly, we can see whether we can get co-infected birds, which is interesting, but we may miss some infected poultry because we don't swab every bird in the market. And so the environmental sampling can fill in those gaps, but the weakness of that is that we don't know, we cannot really tell co-infection. So anyway, that's why we do both techniques, and these sample collection techniques are complementary to each other. And so as part of capacity building, we also have supervisory trips and activities with local partners. So these are just two of the more recent trips in May in Luang Prabang and in Vientiane capital. And so once the samples are collected and they come to the laboratory, we were able to put in brand new platform on how to process these samples and to analyze the data that were generated. And as you can see, with all the activities in red here are new, supported by IPSN. Our current capacity, we knew how to do RNA extractions, and we knew how to do PCR for other viruses. It was not very hard to adapt to avian influenza protocols. But as you can see, after we get a positive result, we were able to put in platforms to that either directly sequence these samples with low CT values, or if they have high CT values, we were able to amplify the viruses in embryonated eggs before we directly sequence them. So this is quite interesting for us and quite exciting. So just a quick summary of the results. We collected over 1,000 samples from the live bird markets since last year, and about 38% of them were found to be AIV positive by screening. We selected representative samples that were positive, and we generated over 40 full-length genome sequences directly from the sample, and also a further 40 sequences from the amplified viruses from embryonated eggs. If we were to look at this data by month, you'll see that we are able to detect avian flu every month, in these two locations. But you may also notice that to the right-hand side, so starting at month 7, month 8, and month 9, there was a dip in the avian influenza virus prevalence in the market, and this is due to and these are because of the months after Lao New Year. So this is where they clear out all the chicken because of the festival, and then they get a new batch of poultry in. We suspect that's the case. We don't really have any evidence to support that even more, but we've seen it in other countries as well, where after major festival, the incidence of AIV drops. And if we were to analyze our results by sample type, we were twice as likely to get AIV positive sample in the environment as compared to animals, so 21% versus 41.2%. But where we were really finding the highest proportion of AIVs was in the wastewater. So this is where they wash the chickens, where they kill the chickens, and also the slaughtering equipment, which is nearby. So we were getting above 50% in these sample types for positive rates. And if we were to subtype them, we mostly got H5 subtype viruses, so about 2/3 of our results of our AIV positive samples were from H5. About 10% of them were H9. We also have about 15% that we were not able to subtype. We have done other PCRs as well against other subtypes, but they did not come back with positive results. And then we have some co-infected samples as well, both from the environment and from the animal, although they were not in very, very high amounts. And if we were to look at mutation analysis for H5 subtype viruses, I'm not going to go through all of them that we found. I'm just going to focus on the one for PB2, so in red. So it's an E627K mutation in the PB2 gene, where we know that if this mutation exists, it's much more virulent in mammals, and especially in humans. And actually, for human infections, especially in Cambodia and in Vietnam that were reported recently, all of them have this E627K mutation. So this is something that we look for. And we also found this mutation, although not in high amounts, but we did find them in our samples as well. So 3 out of 38 sequences. And if we looked at H9N2, we were also looking at similar mutations, but for H9N2, we did not find the E627K mutation. And then if we were to do the phylogenetic analysis of these sequences, most of the H5 that we found belong to clade 2321E, and this is the same as what they found in birds and humans of Cambodia and Vietnam. We also have a minority number of sequences that belong to 2344B, which is the dominant clade worldwide. And the phylogenetic analysis for H9 showed that these viruses belong to clade B471, which is not surprising because it's quite consistent with the sequences from past studies in Laos, Cambodia, and Vietnam. And if you remember at the beginning, I talked I said that we did a little bit of air sampling recently. So this is some preliminary results. We collected 6 samples so far, and and 2 of them were positive. However, we found that at the moment, this is very dependent on the surrounding environment. If the surrounding environment is positive, then we were likely to get positive samples, and if it was negative, then we wouldn't get anything. We have one sample that is of lower CT value that we can try to directly sequence, and one that we need to amplify in eggs, so we're doing that work right now. So we also, earlier this month, held a workshop on, supported by IPSN, to transfer a lot of this skills that we learned from this project to local Lao national partners. So supported by Dr. Eric and Jay at IV Cambodia and Victoria from Wipro. So thank you very much for the support. We're able to transfer our skills to the local Lao partners. And so through this project, we implemented sample collection in live markets. We implemented novel cutting-edge techniques, efficient pipeline for generating these full genome sequences. This is unique in Lao PDR, and we are committed to further doing this work. So thank you very much. And yes, thank you very much to the team. Thank you very much to our local Lao partners and also our partners at the Institut Pasteur to Cambodia. Thank you.